Yo, what's up? It is Sinson Greymind here, and I welcome you to my lab channel. I have an update. I'm going to move to Netherlands for some time, and because of that I need to get rid of some of my samples. Some of them are old contaminated samples from experiment which was not well recorded. The experiment was my try for cultivating fungi, but my samples get contaminated. So now, before I will get rid of it, I will observe them under my microscope. I got Mycena chlorophos and bioluminescent fungi and Cordyceps militaris. Fungi known to be parasitic for some insects like ants, and sometimes they call them as zombie fungi because they can control mind of other insects through the chemicals. It cannot possess other organisms like mammals though, and it will need a lot of time to evolve, so The Last of Us is not happening anytime soon. Yeah, so we are going to look at it. I'll put it here, the microscope, and here we can watch. In this first sample, as you can see, it is contaminated with yeast, with those small dots everywhere. For me, it smelled like ethanol, and also smelled like fruits. We are on another Cordyceps militaris sample. This one smells more like yeast or bread, and again, it is mostly about the yeast colonies. This sample even looked differently by a naked eye. It looked more browny and darker. It also smelled more dusty and almost like soil. Also, as you can see, there are something like bugs. Those are dust mites. I am not sure how they get here, but probably I didn't seal the parafilm properly. And because of that, there could be holes through which they could get here. This sample is with clear agar. It means without any peptone and dextrose. It doesn't have any rich smell. Only what I smelled is fungi, and this agar was already dried. I didn't notice here anything different than nice mycelium. Now we are on our first Mycena chhlorophos SDA sample. Mycena chhlorophos is a bioluminescent fungi, which means it glows. This one smells like yeast, and by naked eye you can see mycelium colonies of two different species. I especially like this one, because it looks like slime mold. And here we have second one and third one Mycena chhlorophos SDA samples. Both smelled like dirt or soil, and both had a lot of dead and alive dust mites. Also, on Mycena chhlorophos third sample you can see clearly a lot of fungi spores. And this is my junk petri dish sample. For first, it was used for slime mold. Then it died, and then mold started to grow here. I keep giving it some oat flakes so they can grow more. And that's everything for this video, and see ya next time.